everyone that worked with BIOLOG to have the chance to discuss some of their questions, some of their interest in the technology, and also for new people, you know, that are exposed to the first time to learn a little bit more about this microbiology-based technology that has different application, as we will see today. So when Stefano contacted me a couple of months ago to discuss this, we thought that it would be a good exercise for everyone to go through an overview of what are the main BIOLOG applications. You know, spend like 45 minutes together to discuss this. And of course, BIOLOG has been around for many years. Some of you may know it. Some of you may be too young to know that, but you know, we're talking about a company that is present since the late 80s doing microbial phenotyping. So there's plenty of application that have been done using BIOLOG. Today we will see some of them. There are others that were left out for simply a matter of timing. But the core will be highlighted today in this presentation that I decided to, you know, to call functional characterization of microbial isolates and community using the BIOLOG technology, an overview. And this is the thing that I would like to touch today, the different topic that I would like to touch today: biotechnology, how it works, how does it work? It's for those of you that are not familiar with it, but also, you know, for some of you that have been using to discover a little bit of the rationale behind it. The beginning, so how BIOLOG actually started into this phenotyping space. The microbial identification, will just talk just a couple of slides about it, and then we will dig into the Phenotype Microarray technology, or the more extensive metabolic characterization of microbes and the main application. And then also we will talk about microbial community functional analysis that you can do with BIOLOG plates and all the options that are currently available. The last couple of slides I prepare just for you to know. I know nobody here is probably interested in those, but just for let you know that BIOLOG, after many years, you know, developing application on the phenotyping space for microbes, that developed also plates that can analyze human cells. And I will just show you a couple of slides just for your info. So let's start, and let's dig into the first application that was developed by BIOLOG. As I said, the company was founded by Barry Buckner, who's currently retired, just retired a couple of months ago in the late 80s. And then it was immediately implemented to do microbial ID, but the base, the rationale behind the technology is basically the same. It doesn't matter if you're looking at ID or phenotyping characterization / metabolic characterization, or if you're looking at functional community analysis, or if you are, you know, looking at a metabolic fingerprint of a human cell. So it's exactly the same, and the core of this technology is, you know, what you see actually in this slide here. Is a 96 well plate, very similar to the 96 well plate that you can get from Corning, from Sigma. Just wells are smaller, and the feature of this 96 well plate is that BIOLOG add a different substrate in each of these well. So you may have, you know, a sugar in well A2, an amino acid in well E3, and so on. So all different well contains a different compound. Each compound is, you know, a standalone metabolic test. And what the user has to do essentially to run this test is, again, very streamlined, very simple. You take the plates that contains already these compounds pre-dried in the bottom of the wells, and then you add your your cell that you want to characterize. It can be, a bacteria, a fungus, a yeast cell, a mixed community, and now, as I mentioned, human cell or even mitochondria. You add them in a standardized inoculum, so each well has the same amount, more or less. That's very important. And then you want to see how they respond in each different well, if they are metabolically active in each different well. And the way you visualize the metabolic activity is through a probe that is a redox dye, essentially. That if the cells in the well are active, they will produce a purple color that is visible by human eyes, and it can be also measured. And this reaction will, as you see in the animation here, will happen over time. The timing of the reaction is different, of course, depending if you're working with microbes or if you're working in mixed community or with human cells. But essentially, after a certain amount of time, you will reach what you see here. So some wells will be having a color, some other wells will have no color. Actually, the picture is a little bit misleading. Some wells can be also, showing a shade of color, not just a yes or no answer. So that could be also a quantitative signal that you can measure. And that's also very important. But essentially, at the end of the experiment, what you get is metabolic characterization, or if you're testing different strains on the same compounds, also what is called a metabolic fingerprint. And this, in fact, actually, is the idea that Barry Bochner had back in the 80s. On the same plate, different microbes will give different profile, and this can be used. This fact can be exploited for creating a technology that can identify microbe. And that's how, the very first application for BIOLOG was created. It was essentially an idea that was already present in literature, and other scientists had. For example, this Dutch scientist L.E. Den Doren de Jong in 1926, working on Burkholderia species, already published a paper showing that you can differentiate, I should say, different Burkholderia species by their carbon source utilization profile, scoring them into positive and negative simply, and then using that as a key to identify them. So it wasn't really a new idea, but the new feature was to parallelize this in a 96 well plate, so having almost 100 different tests with a different level of resolution, and then exploiting that for unique identification technique. And this is these slides here just want to summarize this, how it works. You look at the same plate, same 96 well plate divided by BIOLOG. I will show you in a minute the latest one for microbial ID, bacterial ID. But essentially, you see here 4 different bacteria: Moraxella, Corynebacterium, Streptococcus, Acinetobacter, all run in the same plate type. And you can see that they all give a different metabolic fingerprint, so to speak. The plate has, of course, negative and positive control for reference, but for example, Streptococcus, Acinetobacter, you can see they're almost complementary. The Streptococcus likes the top, is active on the top compounds. Acinetobacter is active on the bottom compounds, but also the other two bacteria, they show a different profile. And using this, exploiting this, and creating a database, then allows the technology to become an effective ID technology, which was very simple. Taking an isolate from an agar plate. As I said, you prepare a standardized inoculum, and then you inoculate the plate, possibly with a multi-channel, so you don't insert any error on volume. If possible, this is obviously very important. And then you incubate over time, and when you get the profile, you run it against a database that BIOLOG has curated and prepared over the years. And then if you have a match in the database for the profile you have, the system will give you an identification. So that's a very simple and streamlined protocol that from left to right, it takes less than 5 minutes to prepare the plate. And then, incubation time is dependent on, as we said, on the organism that you are considering. It's 24 hours for bacteria, a little bit more for yeast, and it can be 2 or 3 days for filamentous fungi. But just to give you a little bit more details before we move to the next application BIOLOG developed that I think is more of interest for you, for most of you, I want to show you, this is the latest plate that BIOLOG developed for bacterial ID. Several versions were developed. Now we are generation 3, and you see there are the plate is divided in 2 parts. 171 carbon sources on the left, divided in between sugars, amino acid, and carboxylic acid, fatty acid, and 23 chemical sensitivity assay on the right, where you have tested for pH, lactic acids, or sodium chloride, or antibiotics. And this is the profile you see, typical of Stenotrophomonas maltophilia. So when you have that microbe, with some degree of variability, you will end up having always the same profile if you're working in the same condition. So this technology was, as I said, was developed in the 90s, and it was quite successful. And still nowadays, big pharma company use our technology for ID, also medium size, of course, and also researcher they do. And this is, I think, because the technology is quite simple, as I show you, the setup doesn't require a lot of skills. You need to have very basic skill of microbiology to do that. But at the end, you get an ID, but not only an ID, you also get biological information. So what is the strain or the cell that you want to identify? What is capable of doing? What are their metabolic feature? Can they metabolize simple sugar? Can they use more complex sugar? Can they use specific sugars, or are they resistant to specific environmental condition? This is a technology that can provide identification, unique identification, but also biological information. And of course, this can be interesting, and was interesting for many of scientists like you, and that asked then BIOLOG to develop more, particularly not only the identification technology that was developed for anaerobic strains, yeast strains, and filamentous fungi, but in particularly there was a lot of request of providing more tests that could basically characterize more in depth the metabolic feature of microbial isolates. And that's what happened in 2003, 2004. Actually, BIOLOG developed the Phenotype Microarray technology, which is essentially an extension of the microbial ID. And you will see in a moment, when I will show you the feature of the PM system, why this is an extension. And this was done, as I said, at the beginning of 2000. BIOLOG started working on this on 2000, essentially, and released the Phenotype Microarray in 2004, I think. That's when I just graduated from the University of Florence, and I had the chance to work on the first BIOLOG machine in the University of Florence with Professor Carlo Viti. And it was the time that high throughput technique like microarrays, you probably remember or you heard about them, DNA microarray, start emerging. So that's what prompted the name of Phenotype Microarrays as this early 2000 taste, if you want, right? But it's a high throughput technique, as you will see. And back then, again, we had genomics going strong. Then transcriptomics was important to characterize isolates. proteomics and metabolomics were all emerging, and now they represent a solid base for, you know, any microbiologist that wants to characterize more in depth their strains for whatever application they have. And I think it was a right moment, because Phenomics was left a little bit behind, and BIOLOG took this niche, if you want, because it is a niche technology, but still able to differentiate itself, mainly because it's a cellular analysis. As I mentioned initially, when you want to start a BIOLOG experiment, you take cells from an agar plates. The cells are alive. So you basically work on cells that are alive, and they need to be alive to show all their metabolic potential. While in the other technology, like transcriptomics, proteomics, or metabolomics, they're really good ones. They are molecular analysis, and you have to essentially kill the cells to extract the protein, to extract the RNA, to extract the metabolites. And so you do some sort of snapshot, and you can repeat these snapshots over time after treatment, whatever, but still you have to kill the cells. While, you know, BIOLOG is a cellular analysis, and this is, of course, you know, more as a motion picture. So it provides a different and complementary angle, if you want, that I think for those times was really relevant. And still nowadays it is, and as you can see by the number of publications that are emerging, and by the fact that we are having this meeting today. So just to show you a little bit more in depth, you know, the Phenotype Microarray is a more high resolution technique. So I think it deserves to explain a little bit more how essentially works and what exactly the dye is going to measure. So we said that there is this a redox dye. There is a tetrazolium-based redox dye. There is a probe for metabolic activity. This slide here shows a cell. You know, it's a funny-looking one, square one, but it could be any cell, really. It could be a bacterial cell. It could be a yeast or a fungus cells. It could even be a human cells if you put a compartment here on the TCA cycle, right? And there is a mitochondria. So essentially, just to say that it will work all in the same way. You know, this is pretty universal as a mode of function. There is glycolysis, TCA cycle, and in the middle, not by chance, the electron transport chain and the NADH. They are circled because that's exactly where the probe takes the electron from and reduce himself and flag the metabolic activity. So, for example, if the cell finds a compound like glutamine in the well, and the cell has the genetic repertoire to bring glutamine inside and metabolize it, there will be an activation of the TCA cycle, in this case for glutamine, and an activation of the electron transport chain with production of NADH, production of electrons, and then cis electron will be captured partially by the redox dye that will be reduced, and it will change from a colorless status to a purple color. The intensity of the color, as I said, is proportional to the level of metabolic activity. More color, more metabolic activity in the cell. And this is very true, you know, even if you use a compound that goes like through glycolysis, like fructose, the end result is the same: activation, electron transport chain, NADH production, electrons that reduce the probe, and production of color. So essentially, these fructose glutamine are just two examples of the entire subcircuit that you can find in the master metabolic circuit of the cell. So you can think about, you know, this map. You know, we all seen this map, right, in the biochemistry books, and we were all scared. But I think you can somehow separate this map, break down this map in several different experiments, and that's what BIOLOG was trying to do with their plates. And each plate, you know, there are different plates to focus on different segments of the metabolism. You will see in the next couple of slides, but in each well particularly, you can focus on one specific part of that metabolism. So you fragment that down, you parallelize, and you have a high throughput test where you can characterize extensively your strain in a very short amount of time. And again, I think the strength is not only the level of data, the amount of data you can get, depending on which is your interested, carbon source or something else, but also is the ease of use of the technology. As I said, the setup is very simple. You take your plate, you add your cells in a standardized inoculum. The cells, they find a compound, a different compound in each well, and they will respond to it depending on their, genetic characteristic and capability or not to metabolize that. So it's a very simple technology, very informative, and that has essentially two components. One component, we discussed about it, are the plates. There are, for Phenotype Microarray, many different ones were developed, and we'll see in a second. And there are in two categories, essentially, plates that stimulates the metabolic state of the cells, and plates that have chemicals that inhibit the cells. We will see in a moment in more details. But the other component is the OmniLog incubator reader. And this is, I think, what made the technology really high throughput, because this instrument that comes with a computer and related software, and you can incubate up to 50 plates inside, so 50. So you can set them in, and the reader will automatically read them every 15 minutes, or you can set it up even every 5 minutes if you want, and then plot automatically over time the signal intensity, giving kinetics measurements of color formation. So this is just before we dig into this is the details on the different plates that BIOLOG developed for the technology Phenotype Microarray. So not only one plate that was used for ID, but several different plates that target different segment or area of the cell metabolism. There are plates that focus on carbon metabolism, plates that focus on nitrogen, simple nitrogen, and nitrogen pathways for dipeptides and tripeptides, phosphorus and sulfur metabolism, pH or osmotic metabolism, and then biosynthetic pathways or nutrient supplement to understand if there is any cryptic auxotrophies. And then these are the plates that test, the metabolism and stimulate the metabolism of cells. And then there are plates instead that have in each well a different chemical that can inhibit the cell potentially. So antibiotics, heavy metals, mutagens, and each of these compounds is present in 4 different concentrations for amine titration. So that's of course a large selection for the user. The user doesn't have to run all the plates. He can choose only the one that he wants to focus in. But if they do, there is a 2000 phenotype essentially that can be analyzed all at once. And again, these are the experiments are easy to run. The OmniLog can accommodate several plates, and then, as I said, gives you the kinetic thanks to the software and the measurement every 15 or 5 minutes. So if you fill the OmniLog, it gets really high throughput. It can give you 5,000 different tests done at the same run. You don't need, again, to run all the plates. You can run only 1 or 2, depending what is the extent of your experiment. But the OmniLog obviously helps a lot, and unlock also this kinetic information, which is really important, because each of the curve that you've seen that the software will generate hinders, a lot of different parameters that can be extracted with a few clicks, and can be very important for the characterization, the deep and high resolution characterization of the strain you want to assay. So for each reaction that is positive, you can calculate the slope of the reaction, the lag time, the area under the curve, and so on. So this is just a general overlook of how things work for the Phenotype Microarray. I would like now to spend the next 10 minutes showing you some notable example on how this was used, just to give you some ideas. As I said, I had to leave many other examples out, and I wanted to keep the main ones for you to have a general idea, but there are many more, of course, and as you can see by the number of publication that comes out, if you just type in Phenotype Microarray, BIOLOG, or metabolic characterization with BIOLOG in Google Scholar. So this is a typical example of strain characterization. So this is Corynebacterium glutamicum that was characterized on the 20 plates that I showed you earlier. The carbon sources, the nitrogen, the osmotic, the chemical sensitivity. Again, you don't need to characterize all of them. To use all of them, you can focus only on those that you are interested in, run maybe 1 or 2 or 3 or only nitrogen. But this was a full characterization for the purpose of devising a better growth media, a selective growth media for Corynebacterium glutamicum, which is used a lot in bioprocessing. So in this test, it was possible to see what they were their favorite carbon source, the one that not only was using efficiently, but also were producing more energy in the cell. And the same nitrogen sources and the same peptides, the osmotic condition and the pH condition, and all the sensitivity that could, and resistance that could make selective media for Corynebacterium very, very possible in just in devising in less than 48 hours. So this is a typical use, and you find a lot of papers where they characterize this, and of course, when after they done their characterization, to compare, also different strains. These are very, very good papers that is almost has 20 years, but it still stands strong for how good the characterization was made in Saccharomyces cerevisiae natural diversity strains, and they were analyzed for dipeptides. Dipeptides are really important in yeast. They dictate how carbon and nitrogen at the same time can enter the cell. The transporter are very different across depending where the yeast is coming from. And this was shown at the genetic level in this paper, but mainly also proven at the phenotypic level, metabolic level, using alive cells. So this is a very seminal paper from Oman et al. That I always recommend, where all the information coming from a selected set of papers can be conveyed to answer ecological questions. But also the technology becomes really strong when you use the metabolic curves that are generated for comparison. Maybe between a treatment and a control, or a mutant and a wild type. And this is important. The kinetic is so much more informative than just reading endpoint. You can see here the example is clear. To control an experiment, so control and treated in a certain metabolism, X metabolism over time. If you read endpoint, they have the same results. If you read over time, it appears clear that there's a huge difference in the lag time and the area under the curve. So it's an instrument, it's a tool that gives you more view, more resolution on this potential difference. And as I said, when you do comparison, this can be very strong. And these slides also reveal why the name Phenotype Microarray was chosen. You can have two different cell line, again, treatment and control, or mutant and wild type, and you run it on the same plate. The system, you put them in the homolog, the system will assign a color to the control, to the curves of the control, assign a color, a different color to the other cell line that you want to compare it with, then overlay them, and where you see yellow, essentially immediately the metabolism is the same, and where you see a color over the other, either red or green, there's higher metabolic activity in that particular cell line to the which the color was assigned to. So again, this is if you remember microarray, DNA microarray, the idea is the same. So you can imagine that it can be very good for comparison of mutants. This is an example of E. coli mutant with a canamycin cassette. You can see immediately what are the effects on the gene, if you targeted the right gene, if there were pleiotropic effects, if the cassettes, inserted in the right gene that you wanted. So a lot of studies, and personally, I also use for this in the publication that I have done in the University of Florence, but also mutant versus wild type, but also when it comes to more high resolution metabolic analysis, and this can be very relevant for modeling, right? Metabolic modeling. You can, for example, compare the very same strain, for example, that was grown in two different physiological conditions. So E. coli here, run at and grown at pH 7 that you will see in red, and run versus E. coli grown, at pH 5. So a big difference of 2 pH would cause metabolic shift, and those were expected. You see a lot of yellow, which mean overlap of the two, but also a lot of color, green, just less green, but a lot of red, meaning that at pH 5, a lot of the metabolic activity were changed or were affected or even shut down. So I'm not gonna highlight everything here, but an overview, it's clear. And then if you want to focus, of course, only on one specific aspect, you can do it. And also, this can be done, pH is not the only parameter that can be changed and then analyzed on a broader scale. You can change temperature and analyze the strain at different temperature and see how they behave. This is very relevant, for example, for pathogen. Like Yersinia has a certain metabolism at 26, but when it's closer to host temperature, some certain metabolites, certain specific metabolism, they activate. And this is an example with 3 different strains of Yersinia that shows only on carbon source. This is an example where you don't need to have all the cells, but you can focus only on a specific segment of interest of the metabolism. Shows already interesting and relevant difference at the basic metabolic level. And you see fumarate, aspartate, malate activating when the temperature shifts. Another nice thing to remember to consider is the technology is very flexible, and you can analyze aerobic strain, but also anaerobic strain using sachet. This is really good, of course, if you're characterizing, for example, strain extracted from sediments or from anaerobic human district like the gut. This is an example with Helicobacter and Campylobacter that they can give their profile in aerobiosis, or if you want to see facultative anaerobes, how they change their metabolism where oxygen is present and when there's no oxygen. This could be another relevant application. And the last one I would like to present on the Phenotype Microarray, again, that is very popular, is the use to establish the mode of action using a tentative compound or a drug that can be using using large screening at different concentration, large screening with increasing concentration of the drug to understand the mode of action of the metabolic level can be done very rapidly in very few days. And this has been used a lot by pharma R&D currently and in the past. And this is an example that shows for a fungus like Botrytis cinerea. And this is particularly focused only on the carbon metabolism, and 5 different fungicides were tested and show different mode of action on the metabolism, shutting down a glycolysis, TCA cycle, total shutdown, or no effect. So This is another very common use of the phenotype microarray, and as you can see, it can be a broad analysis or a more focus on what is particularly of interest, and this can be done at different doses in basically no time. So very powerful and informative, as you can see. Okay, so I think I have 10 more minutes, and I'll try to use this to show also how the technology was employed for functional analysis of microbial communities. So the BIOLOG technology was initially devised to work with single strain, but obviously scientists like you, they saw an opportunity to not use only isolates, but also use mixed communities. And the BIOLOG identification plates were used for this purpose. And you can find a lot of papers that are coming from the 90s or the early 2000s, where identification plates for BIOLOG were used with mixed community, the GN plates or the GN2, GNP, a lot of them. And then BIOLOG decided to produce a specific plate that is called EcoPlate, and I'm sure many of you heard about it. There's a huge body of literature about it. It's a special plate that has these 31 substrate carbon source only, in triplicate, so replicates or different samples can be run. The feature of this is that are more suitable for microbial consortia, because instead of having simple sugars, they have more complex carbohydrate that needs, a more a collaborative, most of the time, collaborative metabolic activity by different species to be metabolized. So there are carboxylic acid again and carbohydrates, but also amine, phenols, and polymers and amino acids that are listed here that can be divided in functional classes and then been used extensively. And the way the plate works is exactly the same as we discussed earlier. There's the compound is pre-dried on the bottom of the well. There's already a redox dye into the well, so you don't have to add it. You can, like you do in Phenotype Microarray or in identification, it's already in there. And you add your mixed community, and then you can have an idea of what type of function across the one that I'm showing you in these slides, what type of function your microbial community is able to do it. And there's a link, on the number of function and also the quality of the function, of course, but mainly the number of function to the functional diversity and the microbial diversity. More function equals to a more diverse community, more functionally active. And this is, something that it can be, linked easily with other technique like sequencing or, TRFLP or DGG to understand how the community change over time in response to a stimulus, etc. So again, there's a lot of literature available about this, and I just wanted to show you that probably some of the best, well, I shouldn't say the best, but the most novel application for ecoplates. Ecoplates can be used with soil bacteria, with any type of community bacteria from plants, from water, from human. And I just want to show that it has been used also with mixotrophic microalgae, that they were able to provide a characterization of them, and also to understand what were their ecologically difference, what they were niche of each of these microalgae that were tested in this very nice paper. All these algae that you see here were tested and then analyzed, as I said, for their functional niche. But also, ecoplate has been used with anaerobic community in sediments, but also in soil and wastewater. But this paper show efficiently that they could be used with anaerobic sediments and merge together the data coming, with as I was saying, with the 6NES sequencing data, and then coming to a more stronger conclusion, because data are complementary. You get an information on the structure of the community by the sequencing, and then you get an overview of the functional capacity or abilities of this community, and see how it responds over time, over different treatments, over different environmental conditions. And you can link also these environmental conditions to specific response in the community, of course, if you do more elaborate analysis as multivariate data analysis. So EcoPlate was one of the solution that was developed by BIOLOG. But again, scientists decided that other plates could be interesting as well, not only the EcoPlate, and you find now a lot of publication where even the Gen 3 plates were used, the PM plates, particularly for those scientists interested in the metabolism of a certain community on nitrogen. FF plates, I didn't mention them. They are specific for filamentous fungi identification, and they have been used to characterize filamentous fungi microbial community only. There's few paper with this, and also AN plates that are developed by BIOLOG for the identification of anaerobes, but they are really efficient in characterizing anaerobic community from for example, the human gut, but wastewater as well, or sediments. And all these different plates, they have different features, and they can. So the scientists, they decided to use it. There are plenty of examples. I'm just going to show you here one. The AN plates was used with the anaerobic community. In this case, we are talking about the mouth, again, human microbiome, but the mouth of individuals that has a disease or do not have a disease of periodontitis, and to see if, yes, you could see a difference if you look at the communities of healthy individuals against the people that had the disease in terms of community structure and 16S. But could you see this also functionally? And this was possible, doing a simple Venn diagram. There were some functional capability of the community that were typically associated only to the disease. And of course, they can represent important data to have, and this obviously then can be translated into multivariate data analysis that shows how the community functionally is represented, what are the metabolites the metabolism, that contributes to the diversity of the two communities. And this also can be integrated later on with sequencing data to get a more extensive view of the technology, the community, and how they respond to the different conditions. Okay, so I think I have only 2 more minutes.
Yep.
And I'll just quickly fly through this, Stefano. Actually, 1 more minute, but just quickly to finish, this presentation. Reiterating that BIOLOG continue developing new application, and there will be more also coming in the near future. But the most recent one was to go, as I mentioned already, and provide the same analysis to characterize human cells. And the very same technology, so 96 well plates in the OmniLog, although these plates are all compatible with any plate reader, the very same technology of a redox dye, an agar plate, a 96 well plate with different substrate can be applied for what we call mammalian cells or human cells, and characterize them metabolically wise, and see how they respond. And this has been used in many publications. This is an evolution of the technology that came out late in the late early 2010, I would say, and has been used, in study cancer, study drug effect on cell, neurodegenerative disorder, immunometabolism, which is really important, and also, stem cell differentiation. The very latest one, and this is my final slide, is the application that allows to characterize mitochondria, mitochondrial metabolism. Mitochondrial metabolism in human cells, focusing only on this part of the metabolism, the TCA cycle, and that is here in red, and that can, provide functional information on how the mitochondria respond to different drugs or different growth condition or in different physiological status. So this is the very latest one, came out in 2018, and as I said, there will be more coming in the future for microbiology, and so maybe next time that we will have a meeting, I will be able to introduce you to something new. But for now, I want to really thank you for your time, for these 45 minutes that on the spot that we spent together. I hope that this was useful for you to prime some potential idea, to understand a little bit better how the technology work. And I, of course, will be happy to answer your question now, or if you prefer, later when we have the question and answer session. And this is my email here. Please feel free to reach out if you, have anything you want to discuss further with me. I'll be happy, to hear from you. Thanks, Stefano.
Thank you, thank you, Enrico, for your very exciting presentation and a very nice overview on the potential of this technology. So first of all, I want to say that we have time for one or two burning questions, if any. Otherwise, we can maybe move on and open the discussion afterwards. Okay, I don't see any burning question now. Just one from my side. So you said very well that the user can have a very wide series of different plates to be selected depending on the target and the scope of the work, etc. But it would be great if you could maybe provide some additional information about the instrument to be used. So, I mean, which advantages could be found using the OmniLog instrument rather than a normal microplate reader that is much more commonly available across the labs. So,
yeah, thanks, Stefano. So, well, I think the OmniLog reader, as you know, has a few advantages over a normal plate reader. I think, although it's fantastic that the plates are compatible with any plate reader, because that makes, if somebody wants to do a small experiment and test the technology, is possible without, accessing an OmniLog. But having the OmniLog, of course, has is pros, and for sure is the fact that you can incubate the plates and read them automatically every 15 or 5 minutes in high throughput fashion. That's evident, but going more in details into this is that sometimes, if you manually read the plates in a plate reader, you may not be available to record all the data, and you may lose some important time points. This is very true, particularly for microbial community analysis, because the reaction, they happen at a different rate. You have a complex microbial community, as you know, and you may leave the lab in the evening that you just you read, and there was a few color developing, and you come back 12 hours later, and there's a full plate with color, maybe also too much developed, and and this is annoying, you know, but it can happen, you know, with microbial communities. The Omnilog helps in this, in getting always the good data points. You can cut the data, you can crop the data, but you're never gonna miss the right reading point, and of course then helps you with the data analysis. You know, has this data analysis software that you can do. You can do extract data, export them, visualize them in a few clicks. So these, I would say, are the three main advantages. And as a user, you may agree with me, or you may also have some other that you want to share with us, of course, since you've been a long-term user of the Omnilog yourself.
Okay, thank you. Maybe we can continue the discussion later.
Sure.
So thanks again. Now it's time to move on with the next speaker, which is Pawel. So
yes, hello.
The floor is yours. So please, you can just share your screen.
Yes. Wait a second.
Yeah, no problem.
So you see everything or not? Now you see or not?
Yeah. Yes.
Yes. So it's everything. Yes. Perfect. Okay, you have to wait a little bit.
No problem.
Voila. Okay.
Voila. Thank you.
Okay, so hello to everybody. My name is Pawel Trzcinski, and because Enrico explained to you more or less everything about BIOLOG technology, I will focus on a more technical approach to the research or to the studies with the bacteria with the use of ecoplates or gen-free tests. So, okay, you probably know that, but BIOLOG is the system based on measurements of microbial dehydrogenase activity in the presence of different carbon sources of different chemical compounds. And from this measurement, we can obtain a phenotype, which could be used for the identification or for the characterization of the microorganism or for characterization of the community of the microorganisms. Yes. So there are two types of BIOLOG system. The first one is the semi-automatic microbial identification system with microstation. So this is it. The second one is the Omnilog system. So this is the microstation system with this huge incubator with camera inside. Yes, and Enrico told you already that with this camera you can measure the color development in time. So you can obtain some enzymatic kinetic. Yes. So there are four main tests of BIOLOG designed for microbial identification. So first is for aerobic bacteria, so gen-free. For anaerobic bacteria is AN. For microscopic fungi, so for microscopic fungi are GIST and filamentous fungi, and for GIST identification are IT test. But I would like to focus on gen-free tests. Okay, Enrico showed you that already. So this is the composition of the chemical reagents used in the gen-free microplate. So first of all, to perform studies with the bacteria with the use of gen-free plates, we need to prepare a sample in the right way. So first of all, we need to obtain the pure culture. This is a silly thing, but this is very, very important, because sometimes we will obtain some strange results because we use mixed inoculum. Yes. Then we have to choose the proper agar media to obtain biomass for the test. This is the next very important thing, and I will show you why this is important in the next slides. So then we have to differentiate the bacteria. Yes, we need to know if they are gram-negative or gram-positive, because some procedure need to, okay, for some procedures we need to know that. Then we have to choose the proper protocol, and this protocol is mainly depending on the cell wall properties. So if the bacteria is gram-negative or positive, then plate inoculation and incubation. Yes. So first of all, obtaining the pure culture. So the streaking is a very common technique, but sometimes we will obtain a mixture of the very similar colonies. The other technique is the spread plating of the bacterial inoculum. So we have to dilute this inoculum several times, and okay, we'll obtain something like this. And okay, for example, here you have you can see the spread plating technique, and in the middle there is the colony, which is which differ from the another. Yes, so with the spread plating we can observe that things. On the streaking, we can okay, on the streaking probably everything will be okay, but here there is a colony with another morphology. So sometimes we will not be able to observe some very small contaminants. Yes, so I think that this is very simple and basic thing, but obtaining a pure culture is crucial. The next thing, we have to choose proper media for obtaining the biomass for the test. So the default medium is BIOLOG universal growth agar. Yes, with or without the fibrated blood. In my career, I never use the BAC agar with blood, but I don't know. Perhaps for some clinical samples, the blood is crucial. Yes, if you don't have the BAC agar, so the BIOLOG universal growth agar, you can use TSA. Yes, for any brand, but there will be some differences between microbial metabolism. So if your bacteria will not grow very well on BAC agar or on TSA agar, you can use chocolate agar. Yes, the chocolate agar is containing more nutrients and more complex nutrients. And so look, I don't use this agar because I work with environmental isolates, and they okay, they in majority they do not need a very complex microbial media. And the last one, so, is the R2A agar, and in my opinion is very good for environmental samples. Yes, for example, if you would like to study metabolism of some streptomyces, the R2A agar is very good because on this media, the streptomyces will form a lot of exospores. Yes, and you can make a very nice inoculum with the use of the exospores. Another thing is that we need to use for the test the fresh culture. Why? Because we have to prepare the inoculum with some proper turbidity. If we use old culture, we'll obtain the turbidity, but the inoculum will consist a lot. Will consist some dead cells. And okay, this could have an impact on the results. For example, here you can see the comparison of metabolic activity of Panibacillus polymyxa cultivated on back agar and onto R2A agar. Yes, so the green part of the plot or the graphics is from the metabolism of Panibacillus cultivated on back agar. The red one is on R2A agar. So yellow parts are the common for the two metabolisms. So here we can see that the microbe cultivated on the nutrient poor agar has some is more active, for example. Yes. So this could have an impact of the results, and I will show you in the next slide. So here we have another example. So the difference between metabolism of strain Pawel and there are some differences. Yes. The strain cultivated on TSA agar will have more. Okay, will be more, I don't know, more active. Yes, more active. So we'll obtain different results. Yes. So another basic thing, so differentiation of the chemical or physical properties of the bacteria cell wall. So we can do a gram staining or similar test like KOH or how the bacteria will grow in the presence of some antibiotics. So the next very important thing is the choosing of the protocol for the identification. So we have 4 different protocols. So protocol A is the default. I can say that it's the default protocol or universal protocol, and is mainly for gram-negative bacteria. Protocol B is for gram-positive bacteria, but you can also find information that is mainly for gram-positive spore-forming bacteria. Also, we can use this protocol for strongly reducting or capsule producing gram-negative bacteria like Areomonas or Vibrio. Yes. Protocol C is for microaerophilic capnophilic bacteria. So capnophilic means that they need more carbon dioxide in the atmosphere. Yes, and microaerophilic, okay, they need lower amounts of oxygen. Nocardia can be also identified with the use of protocol B, but this depends on the strain or on the species. Protocol C2, I very rarely use this protocol, but is for bacteria which need more complex medium. Yes, or more complex medium, or they are oxygen sensitive and they like higher amount of carbon dioxide. Yes, and below you can see the examples of the bacteria. So some common problems with the suspending of the bacterial biomass, because for the test we need homogeneous inoculum. Yes, but for some bacteria, for example from Bacillus subtilis group, for example here and here, there will be some problems because they produce clumps. Yes, so the biomass collected from the medium will produce the clumps, and you can see clumps here. The solution is simply you have to remove only the clumps. Yes. But if you put this, if you transfer this, if you transfer inoculum with the clumps into the well with some carbon source, it could potentially give you some false positive results. So I told you that the choice of the proper inoculum of the proper protocol is crucial, and you have here some examples. So Lysobacter is the bacteria which is not in the BIOLOG database. So at the start I have some problems with the Lysobacter because I don't obtain any logical phenotype. So I use the second protocol, and I obtain some different results. So, for example, if you put some information about the phenotype of the microorganism in the article, in my opinion, this is very important to inform the reader that you use this kind of media for cultivation, you use this kind of protocol, you use some you incubate the plates in some temperature. Yes, this is very important because I read some papers and the author did not give that information, and as you see here, this is very important for the obtained phenotype. Another example. So the strain V3, I use the different protocols. Of course, I obtain the same results of the Vibrio, but there are some differences. Yes, so in that case, we have the same identification, but sometimes you can obtain different identifications. So, as I told you, this is important to use proper protocol. So the plate incubation. So default temperature is 33 degrees, but you can change this temperature from 26 to 37. But you can also incubate the plates in the other temperatures. Everything depends on your studies. Probably, I think that Enrico showed you in his presentation that there are some changes in the bacterial metabolism when the plates were incubated in different temperatures. Another thing, atmosphere. So Enrico told you that already, that you can also measure the activity of the bacteria in different type of atmosphere. But for identification, the BIOLOG company provide some information so about the type of the microorganism, which protocol you have to use, which type of medium, atmosphere, and temperature. Yes. So probably you can find this in the internet. Really, I don't find that information on the BIOLOG website, but probably I don't. So probably I have my internet skills are not good at this moment. So this is everything you will need for performing the test. Yes, here you have everything to perform an identification or characterization of the microorganism. If you need some, if you have to change, if you would like to change atmosphere, you have to use some an aerostat and the kits for generating the proper atmosphere. So the interpretation of the results. If you done everything, if you do everything correct, you will obtain some identification. So you have So identification. So here you have 3 different index. So probe index, so probability of the successful identification. The value range from 0 to 1, where 1 is 100% of probability of correct identification. And this index is mainly used to inform the reader or somebody that, okay, I identified this isolate, and for example, for 81%, this is the Bacillus polymixa. But for the system or for you, the SIM index is much more okay. The SIM index have more value, yeah, because it indicates how well the sample is identified. So it has the same value, so the value range from 0 to 1, and the system, the BIOLOG system, okay, for identification, the BIOLOG system, wait a second, the BIOLOG system compare the results from your microplate with the database, and the profile has to be has to exceed a threshold. Yes, so a minimal threshold for species identification is 0.5. Yes. Distance is another thing I will show you later. Or for example, distance. Distance is an index which is calculated on the base of mismatch reactions. So, for example, for this isolate, you have 2 mismatch reactions. So the distance is 2 with 2 and something. Yes. And okay, I will. Okay, so here you can see some information from the reader. Yeah, this is then an absorbance. For some studies, this is very important, but for simple identification, this graphic is better. This is the simplified graphic that which indicates that, okay, my microorganism is able to metabolize, for example, this carbon sources, this carbon source, and this carbon source. Oh, and I forgot to tell you that we can see 3 different reactions. So white is no reaction. Violet violet color is that microorganism metabolize some compounds, and white and blue color, this is the borderline reaction. So this reaction is very weak. Okay, I told you that on the result screen you can observe you can obtain some information from the microreader. So you have information from 2 wavelengths. Yes, the first, so the wavelength of 590 nanometers is correlated with the color development, and could be connected with the metabolism. Yes, so how strong the microbe or microbe community will metabolize the carbon sources. The second one is correlated with the biomass development, and this is this could be useful if you, for example, develop a medium for biomass production for your isolate. Yes, so this could be very useful. Sorry, something. Okay, yes. So interpretation of the results. Sometimes the system will give you this kind of information that your isolate is probably in this case Bacillus fumilis or sapensis. Yes, this is because these two species are very similar, and with the use of this phenotype, the system is not able to recognize it, but it can tell you that okay, this is this one species or the second one. So you have to so in that case you have to use another test for differentiation. I don't know, some extra biochemical test or sequencing or something else. Okay, okay, the next thing. Sometimes we have two different identification. So this is the same isolate, the same turbidity, the same medium. Okay, this is the same plate, yes. And after that 48 hours, we obtain this kind of identification. And after 45 hours, we obtain this kind of identification. And to be honest, these two microbes are not similar to each other, yes? So what we have to do? Sometimes some simple differentiation like microscopic preparation, or we can use some molecular biology techniques, so sequencing, for example. So common problems. So first common problem is the false positive reaction, so false positive negative control. So what we have to do, the system inform you about everything. So first of all, you can try to use lower inoculum or to change the protocol. In this case, if we start with protocol A, so you should use protocol B. Yes, for example, this bacteria is Klebsiella oxytoca, and it commonly gives this type of false positive reaction. Another problem, no ID. Sadly, the BIOLOG system has some okay, the database is huge, but it's not okay. It not contain every phenotype of every bacteria. So sometimes we'll have no identification. So we have to use another thing, another method for identification of our isolate. So Enrico told you that already, but I will repeat that. So we can use the BIOLOG or OmniLog system for another things, because this is a tool, yes. And how we use the tool, this is our thing. So first of all, we can compare the kinetics of the metabolism. And why we can do that? Because we can observe some mutation or some changes in the phenotype. So for example, we use some Klebsiella oxytoca for test for enhancing the growth of the plants. And after 2 weeks, after inoculation, we waited for 2 weeks. Then we collect the samples, re-isolate the bacteria, and we found that we have two different phenotypes. So one is perfectly matched with the reference strain. The other one has some changes. Yes, he lost some ability, and in this case, he lost ability to grow in the presence of 8% of natrium chloride, for example. Another example, in Excalibur project, we use Panibacillus polymixa strain K16. So we were able to re-isolate some of the Panibacillus from the growing medium or from the substrate, and we were able to compare these isolates with the reference strain, and we found that okay, in the pit medium we have the same strain, so we obtain the same kinetics of metabolism, and we also obtain the same with the same DNA profile. But when we re-isolate the Panibacillus from the soil, some Panibacillus have the same DNA profile, or they are okay, they match the same DNA profile, but they have different phenotype. Yes, and I think this is a very useful tool. The BIOLOG or OmniLog is a very useful tool to compare, for example, the strains or isolates. Another thing, I told you that earlier, but from the data from the BIOLOG system, you can develop the medium composition for the multiplication of the specific strain. Yes, for example, here we have Pseudomonas fluorescens, and this Pseudomonas should be grown, for example, with glycerol or gamma aminobutyric acid as a carbon source, because okay, this is here and here, because the dextrose is not very well metabolized by this strain. Also, the medium should be buffered at pH 7 because the metabolic activity will be lower in the pH of 6 or 5. Yes, another example. Yes, so we can develop the medium for the recovery of the bacteria or for multiplication. So the same, this is the same case as with the Pseudomonas. So we know that Panibacillus likes dextrose, but we can also use another carbon source. So we can use in the multiplication medium just small amounts of glucose to start the fermentation process, and then we can use another carbon sources to for longer time fermentation. Yes. So in this case, we can start with the glucose and then add malic acid or glucose. Malic acid is here. Fructose is here. Gluconic acid is here, for example. Okay. So now I will tell you about the phenotyping of bacterial communities. So the phenotyping could be used for single strain or for the communities. And for these purposes, the BIOLOG company developed the ecoplates. And the ecoplates contain 31 carbon sources repeated 3 times. So as you can see, this is the one part of the plate. Here you have second part and the third part. Yes. So in the Excalibur project, we use one procedure for phenotyping. First, at the first step, we have to estimate the soil dry mass. Why this is important? Because soil from one part of the field could have more amount of water. Yes, and for example here, different. I'm sorry, one more time. So for the test, we need to use the same amount of soil dry mass. Why? Because the results are not easily, it can be not very easily scaled. Yes. Yeah. So we have to use the same amount of soil dry mass in the soil solution. The next thing is dilution of the soil. In Excalibur project, we use the dilution of 1 to 1000, but everything depends on your studies and on your samples, and I will show you that a little bit later. Then we inoculate the test plates, 100 microliter plates, but you can feel free. Yes, you can use another volume of inoculum. In the Excalibur, we incubated the plates for 72 hours in 26 degrees, but of course you can use another temperature, another incubation time. Then estimation of biodiversity and activity, I will show that later, and we can modify this, for example, these two steps. Yes, so dilution of the soil suspension. This is easy to prepare, for example, and the soil solution contains a relatively high amount of microorganisms. But the cons is that the soil solution will also contain high amount or some amount of impurities like soil particles. Yes, and these soil particles would give us some false positive reaction. Another thing is to centrifuge the soil suspension. We'll obtain suspension which is free of impurities, but during the centrifugation, some of the bacterial cells and soil particles with bacteria attached will be lost. Yes. Another thing is the decantation. So this is also easy to prepare, and obtained soil solution will be also free of impurities, but the decantation will take some time. In this time, the microbial population could be changed a little bit, for example. And during the decantation, also the bacterial cells or particles with bacteria attached will be lost because will fall down to the rest of the soil. Another thing, you can in Excalibur project we use distilled water, but you can also use another thing to suspend your soil sample, like saline or some buffer. This everything depends on you. So you can use also different sample dilution. For the soil purposes, we use dilution 1 to 1000, but for some heavy contaminated soils, you can use lower dilutions like 1 to 10 or 1 to 100. Everything depends on your sample. So calculation of the activity of the bacteria. So the microbial activity is calculated in the manner more or less connected with the estimation of enzymatic activity. So Avenger well color development, AWCD index is okay. It's very easy to calculate. So we have to sum the optical density of the wells and divide it by 31. Why by 31? Because we do not use the result from the negative control. Okay, and there is how this optical density of each well is calculated. calculation of biodiversity is a little bit more complicated, but is also easy. Yes. So the index is will differ, but everywhere you will need to calculate the optical density of individual well, and with this result, you can calculate this PI index, and with this PI index you can calculate Shannon-Weaver index. Of course, there are other index in which you can use the results from the absorbance. For example, the Brown index is also very similar to the Shannon-Weaver index, but okay, everything depends on you. But Shannon-Weaver index I think, is most common. Then interpretation of the results and data processing. So this is an example of the results. So we have a lot of numbers, but for example, in this kind of studies, common to see some spreads in the results. So for example, here, and here, you have a different results. So you have to put some, so you have to spend some time to process the data. Yes. So here we have to use not 0.4 but 2 and something. Yes. But everything depends on the negative control. Sometimes the negative control is very high in one field, so we have to check, yes, if this high negative control has impact on the others, readings. Yes. Another thing, we have some splitting data which cannot be easily corrected because we have something like 0, something like 0.9, yes, and this result. So first of all, we have to check the negative control. The negative control. If negative control is more or less the same in every field, well, we have to leave these results. But this is true when you use soil suspensions. Soil suspension. But if you, for example, if you use wait. Okay, when you use soil suspension from the centrifugation, probably you will not obtain this kind of errors. Yes. Another thing, which soil dilution I would have to use for estimation of bacteria activity and biodiversity. So I use here the results from the activity and biodiversity of soil stored over 1 year in the refrigerator. So this is okay. At the first look, this is very strange because in soil suspended in 1 to 10, we have a very low bacterial activity. Yes, and this activity will grow with the soil suspensions. This is why. Because we have strong background, so strong reaction in negative control or strong absorbance. Yes, because for example, this absorbance could be from soil particles. So if we do not know which kind of so if we work with unknown types of samples, this is a very good thing to do some tests and to estimate the dilutions to be used. Yes, for example, we work with the water from the aquaponics, And we use for the water in aquaponic system, we use solution dilution of 1 to 100. But for the soil, it's 1 to 10, 1 to 1000. And for the peat substrate in the greenhouse, we use 1 to 1000, for example. Yes, so the soil diluted in the ratio of 1 to 9 is, so we have the high problem with impurities. But another thing, if we dilute our sample too much, we'll have the problem with repeatability. Yes, so here, so the data from here is other from the data from here and from here. So you need to put in mind that you have to choose between a dilution and between the dilution and between the soil impurities. So you cannot use soil diluted or sample diluted too much, but you cannot also use the sample diluted with too small ratio. Yes. Okay. So the conclusions. So phenotyping with the use of gen-free plates or eco plates or any other plate is a tool for environmental studies. So for example, you can compare the impact of some different stressing factors. It's also good in biotechnology. So for example, you can develop the medium, or you can optimize the process or for the multiplication. Or, for example, the good thing in the studies in microorganism metabolism. Yes, so we can track the changes caused by some mutation on environmental pressure. It could also be successfully used for identification, but in our laboratory we use two different. So we use two approaches for identification. We use molecular biology and the BIOLOG system. Yes. So thank you very much for your attention.
Thank you, Pawel. Thank you very much for your great presentation, a very extensive overview on the bacterial based application, both with individual identification and community analysis. So thank you so much. I think your presentation has also been complementary to the one made by Enrico. So I hope the participants now had clear, I don't know, but exhaustive overview on this technology. So first of all, I'd like to hear from the audience if there are questions, comments, or whatever. And I'd like to start from Anna, because she said that she has no experience with the BIOLOG system. So I wonder if now you got some more information, if everything is clear, if you didn't understand anything, or if you have questions.
Yes, thank you very much, both presenters. It was a very informative, very, very interesting technology as well. I will introduce myself, so then you have a background story, so then you know why I'm here. I work at the Natural History Museum. I'm a research fellow in geomicrobiology, looking at development of bioprocesses for metal extraction and metal recovery. So the group of microorganisms that I work with are chemolithal autotrophs microorganisms. So they get energy from inorganic materials, and most of them also get carbon from CO2. So they fix the CO2. And then my question to you is, could you use your technology, the BIOLOG technology, for identification and characterization of these type of microorganisms, of extremophiles?
I guess this I have to answer this question, right? No, Anna, thanks for introducing. Actually, that's a very interesting field of research. So at the moment, as you can see, the identification technology devised by BIOLOG, it's based on the consumption of carbon source for the generation of energy. So we're talking about heterotroph that you can identify in a reasonable time, right? Because identification methods, generally speaking, you need, of course, to have a response, like a results within, let's say, maximum a couple of days, 2-3 days. And I assume from the knowledge that I have that the strains that you are using, they may take a little bit of time
They
to grow.
take longer, yes.
Yeah, because I work with autotroph once, nitrifiers, and it was you really switch gears to lower gears when you study autotroph and grow them. So for identification, I wouldn't think so that it's possible to do it in a reasonable time frame. To characterize them, there was some discussion, a very vivid discussion in the past about this. So technically, you can, if you allow yourself, as I said, a good amount of time for the plates to develop a metabolic response. And this is sometimes is not compatible with, for example, higher temperature, because then they grow in liquid, the evaporation change the volume of the wells. So, but this is some aspect that they may hinder this. But if you, for example, set up a plate with the proper mitigation for evaporation, use a redox dye, because BIOLOG has different redox dye. There is a very sensitive to electrons, so requires less electron to reduce. In theory, is possible. Then you will have to make sure that you provide to those microbes that you want to characterize all that they need. So that they need CO2, then it needs to be in an incubator with CO2, right? And if they have any other needs of cofactors, these cofactors, they need to be in the plate. So theoretically, it is possible. I would assume that you would need a little bit of optimization to do that, compared to a standard protocol that we discussed so far with heterotroph or mixotroph, so to speak.
Yeah, but also the database as well, because if it's focused more in heterotrophs, you wouldn't have many candidates in a database.
You say right, Anna. Yeah, that's the database is purely on heterotroph, and those are species that generally you can find in the environment. So fast growing. If you want R strategies, there are some slower growing that Pawel was highlighting. Yes, but we are talking about with autotroph, very slow growing. So no, you can build, if you are patient enough, you can build your own database of characterization, but it would be something, definitely only for characterization, not for identification. Identification, I would rather go for molecular methods myself as well.
Okay, thank you very much.
Oh, no problem. Thanks for your question.
Thank you so much, Anna. Very clear your question, very prickly point. You touch immediately very, because in these presentations, we presented the most standard situations, the most commonly applications of these things. So in your case, yes, as Enrico said, I guess you should start with an optimization step, preliminary step, and then, but yeah, it's possible. I remember, I don't know, maybe you remember Enrico as well, some colleagues from CREA, they worked with very slow growing bacteria, lactobacillus, bacilli, something like that, and they took a lot of time for the optimization of the protocol because they couldn't apply the standard protocols of the BIOLOG. So it took months. But at the end they have their own protocol, and this is essential for them because they need to identify. I don't remember which strains of this lactobacilli for cheese production, and these strains were genomically speaking identified, but the same, despite the genome is always the same. When they took these strains, their phenotype was different. So the results in terms of cheese quality was different. So they needed to add the BIOLOG identification method to the genome in order to classify, let's say, the organ. And it was crazy for that. I still remember how could we do? But they did it. So I'm sure you can do that.
It is a really neat technology. So I was like, oh, am I going to have to change fields just to use that?
Well, yeah, sometimes it's just a matter of time. Sometimes it's just a matter of a good idea.
Just
because,
optimizing, yeah.
Maybe
trying. A next generation BIOLOG.
Next generation, yes. I'm pushing on Enrico for an update, an improvement of the instrument and the technology.
Flavia has her hand up.
I'm sorry, Anna, what did you say?
Yes.
Flavia.
Oh,
Flavia.
I just wanted to add something to Anna's question, since I think that as far as I remember, the last software that BIOLOG released allow also the building of a proper of a personal database, so you can record your own standard plates if you have some. And this is one point. The second point is that, for example, I used some Phenotype Microarrays containing, for example, some sulfur compounds, and some can be used only for some single metabolites, and you can also add some metals, given that you also use some bare plates as a negative control to test if you have color formation just for chemical reduction issues. So if you exclude that some color formation is due to chemical reactions, not for the presence of bacteria, you can still try. And maybe a first try can be done with the echo plates, just using one strain, because you have, you know, some substrates. You can see if that environment is good for your microbes. You can also keep the plates for longer time, just keeping them into envelopes, for example. They don't dry out easily. And you can also add some other compounds like glycerol. If you are sure that they are not using glycerol as a carbon source, because they don't use glycerol as a carbon source, you can use it to standardize also and to keep the environment good for microbes that need some kind of osmophilic environment in the plates. So
not all of them are autotroph. So I do have a few bacterial cultures that are heterotroph. So maybe I could test with them first and see.
And also you might test the community. For example, if you have some other microbes growing with the same strains you are studying, and you know that there are some more growing together, you can test the response, the metabolic response of these groups of microorganisms. Maybe they are not so many, but you can test them in ecoplates as a community, and this could be an interesting something to add. Because you have a functional response. It's not just a number of strains. It's really a functional answer.
Yeah.
Okay. I agree with the Flavia. I think one thing that I probably didn't highlight enough in my presentation due to time is that the technology is quite flexible, and it is really up to the people using it to modify it to even to larger extent. I remember maybe Stefano, you remember the scientist from the US, Terry Hudson. He was in a couple of conferences with us with the Phenotype Microarray. He actually, it's hard to forget him exactly. But anyway, he was working on anaerobes, and the beginning, the Phenotype Microarray didn't really have a protocol for anaerobes, so he came up. And you can still find this publication. He came up with an idea of adding this oxidized form of sulfur in the plate, and then putting the plate in anaerobic bags, and this sulfur would be reduced, during the metabolism of these anaerobes that he wanted to characterize, and would form some insolubles precipitate of black color. And this would be used as a indirect indicator of metabolic activity and / growth instead of the redox dye. The redox dye wasn't suitable back then to do this in Phenotype Microarray plate, so he devised, this, if you want, different level of probe. He knew about this because he knew about these strains' metabolic features, so he could devise something similar. So at the end, just to highlight that as Flaga said, you can make modification as many as you want, as long as they make sense and they are compatible with the physiology of the strain you want to characterize, and you're probably at least gonna get information on biomass, if not on metabolism.
Yeah, thank you very much.
Yeah, thank you. So any other questions from the audience? I don't see questions in the chat, but I'm curious to hear Jansha Lamovsek. She wrote a message saying that she's going to get a new macrostation to analyze microbiome, metabolic profile. So did you get some useful information about the possible application of this technology, or is everything clear? Do you have tips, questions?
Oh, hello, everybody. Can you hear me?
Yeah, sure.
Okay. Thank you so much for this presentation. I really enjoyed it. I got a lot of interesting information.
Okay.
I like this last presentation most. And I thank you. Will there be a replay or not? Of
Replay?
This? Yeah, of this.
Once again.
So
yeah.
The presentation is recorded, so you will find that we will send it the link to you all, so you can check it again. So if this was the question.
Yes, this was the question,
okay,
because there was so much information.
Yeah,
it was impossible to make notes.
yeah. No problem. You will get the presentation. You all, of course, will get the presentation as a PDF, and also the recording will be available. So you will be able to re-listen any critical step you may have missed or something. So don't worry.
Thank you so much.
Thank you. I've got a question for Enrico. Maybe we talk about redox dyes, and this is a very critical step of in all senses, considering both the identification approach or communities or regard. So I make this question because as a user, several times I had some issues in the selection of the best, let's say, dye mix or redox, considering that different strains prefer some dyes rather than others, that fungal strains sometimes consume dyes as a substrate, and so it's a tricky point. And I wonder if the BIOLOG has developed some official guideline, some kind of guideline or protocol, as you did for the preparation of the plates or the inoculum for different kind of organisms. So I wonder if you have some news about that, or what can you say about this?
So there was, well, first of all, I think you highlight a crucial aspect of the experiment. Yeah, the dye choice is really, really important, and BIOLOG do assist, you know, with the main strain, but the problem is when you have to maybe characterize a strain that BIOLOG never characterize, and this is happening more often, and you need to navigate by yourself, you know, the different possibilities. So there was an idea now recently to develop like a plate that could choose could help you in the dye selection, or a plate that you don't necessarily have to use entirely in each, but you could use several times, like sort of the eco plate. So this is something that is under consideration among the new product that BIOLOG can release, but because we had the request for you know, for the fine tuning sometimes, but even for the gross setup of new strains. But in the absence of that for now, the suggestion is, well, first of all, I say contact BIOLOG, because you know we may have, you know, someone that we know has used that is publishing that paper, you know, as a, you know, word of mouth can give you some indication that could be helpful. So contact BIOLOG is always a good idea if you feel lost. But if you are navigating this by yourself, you know, the redox dye, they do have different redox threshold. Essentially, they differ by the redox threshold. By the facility, they can get reduced. So before we're done, I was mentioning the use of very sensitive dyes. Some of the dye that we have, and this is information that BIOLOG can share with you if you contact BIOLOG, is not proprietary. Some specific dyes like the dye G, dye mix G, dye mix D as well, and to some extent dye mix H, they are more sensitive to electrons, so they reduce more easily. So for example, if you are working with a strain that grows slowly, you can choose them. And a strain there is, you already have the information, they're not that very metabolically active. I would aim to, you know, to one of those belonging to the very sensitive dye pool. On the other side, if you're working with a strain that grows well, that is very active, grows well, I'm saying, you know, you put it overnight on a liquid culture or an agar plate, and the morning after is ready stationary phase, then I would rather go for, you know, dye mix A, dye mix B, that are more difficult to reduce and may give you, may avoid the formation of a negative, like a high background signal.
Mm-hmm. Yeah.
And then there's the dye for fungi, which is dye mix E. You said right, it's sometimes fungi, they do use it. So we have protocol as a substrate, we have protocol that can help without the, you know, you can use it without the dye. But that's where, again, the Omnilo can be useful, because you can see a color formation in the early phase, and then, which is metabolic activity, and then the fungi, the more they grow, the more they will utilize it, so the color disappearing. So that could be useful, you know, to have the Omnilo get measured for you, even you know, obscure times, and then you can at least have an idea that that is happening, and analyze your data accordingly. So, but yeah, dye selection for now, I'd say contact BIOLOG. There's a document that gives you the threshold value for the dyes, then maybe, you know, it could be a good chance to have a discussion with someone from BIOLOG that could tell you, oh, you know, we have a customer right now doing exactly the thing that you're doing in I don't know, in Japan, and we know where they're having good results with Dimix B. So start from there, you know. So my recommendation would be this in the waiting time of, you know, a plate that could help you.
Okay, thanks a lot. So okay, another person or other persons I'd like to hear are participants external to Excalibur, like for example, let me see, maybe Andrea Ceci. I don't know if you, or Veronica. I wonder if you have experience with the BIOLOG, if you have the instrument, and which kind of applications are you working on? I don't know.
Hi everybody. I have worked with BIOLOG with some fungus filamentous fungi, and we have tested this species in presence of different compounds. In particular, we have selected one saprotrophic fungal species using FF plates, and we test in the presence of lindane and other isomers of ACH. And also in different combinations. We have also tested the solvent used for the preparation of the solution, the standard solution. Moreover, we have tested the same species with these compounds, organic compounds that are actually organic pollutants, and we have also integrated this with some concentration of metal compounds, vanadium compounds in particular. So we have also tested the co-contaminations. We have found that there are in some cases inhibition, of course, of some pathways, and in other cases we also have found possible stimulation of some pathways involved in the degradation of in the use of these substrates in FF. We have worked with Flavia Pinsari, so we have experience in these fields of this application of bioremediation. And this was very important to found some pathways that can also be involved in the biotransformation of compounds. So it was possible to hypothesize metabolism, for example, of compounds in the presence of specific substrates such as galacturonic acid, for example. That can be one of the compounds involved in the transformation of hexachlorocyclohexane or other xenobiotics compounds. And so it was very interesting, this approach.
Okay, thanks. And Veronica is working with you? Or...
Yes, I'm working with him, and I collaborated with Flavia, and I've used BIOLOG only once to make the metabolic profile, the phenotypic profile of 2 fungal strains. and we use the FF plate.
The FF, okay.
Yeah, only FF.
So if you work with Flavia, so you both are in good hands. Only with fungi. So tomorrow will be the most dedicated presentation on this kind of organisms. So I hope I'm sure you will enjoy the presentation of Flavia tomorrow. Okay, thank you. I got a couple of quick questions to Pawel. The first question regards the preparation of the inoculum for identification of strains. This is not the most the application I use the most. So I wonder, in base of your experience, which is your opinion? I'm talking about the preparation of the strains to using minimum media or maximum growing media, because I read some papers where minimum media is recommended because the strain should be put under harvest conditions in order to provide the best metabolic potential, and other papers reported that they had better results when they use strains prepared using a growing media, a maximum growing media, where the strain could be obtained in the best metabolic conditions and ready to be inoculated into the plant. So I wonder, in your experience, which is your opinion about this this step, this?
Okay, so look, so default BIOLOG medium is nutrient rich, for example. Yes, so so BIOLOG, okay. So this nutrient rich medium is good. For example, if you work with Pseudomonas or with how to in another way. So everything depends on the strain and on the source of the isolate. Yes. So when I work with my colleague from the medical university, he told me that clinical strains mostly have to be cultivated on the nutrient-rich medium, for example. Yes, but in the environment, with in the environment, the bacteria will very rarely will grow on the nutrient-rich soil, for example. So so for the environment strains, I prefer nutrient-poor medium like R2A agar. Another thing is on the nutrient-rich medium, some streptomyces, no, almost every streptomyces will form very dense colony, and this colony is okay. It's hard and dense, so you will be not able to take the biomass on the swab and suspend this biomass in the inoculation medium. So, in my opinion, for environmental strains, the R2A agar is the best. Yes, but BIOLOG agar is also good. Yes, because okay, it's default medium, but not in every case. Yes, so I think this is that okay. BIOLOG company provide you with the several different media. Yes, you can find information that this medium is good for this type of microbe and for other. Okay, in one sentence, for me, the nutrient-poor medium is the best for environmental studies.
Yeah, because considering that from isolates from soil usually are unknown, so you don't or maybe you can isolate strains that you are not very expert about, so you don't know exactly the airborne sources they prefer, or the composition, the optimal composition of the growing media. So it's difficult to provide also. If you're using a very well-known strain, maybe you can easily prepare minimum growing media with the exact requirement that they need, but otherwise it's better use the other approach.
Yes,
and
yeah. I'm sorry. Yes, one more thing. For example, the oligotrophs, so the bacteria which grow only on nutrient-poor medium will not grow on the back agar, for example, or will form very, very small colonies. So, okay, if you have options to test several different media, you can use this. But for example, when I cultivated the Pseudomonas on king bee agar medium, so this is very popular medium, and then I tried to identify this bacteria cultivated on the king bee medium. So I obtained the phenotypic profile but I don't have any identification. Yes, so I obtained no ID.
Okay. And another question, very quick, is related to the identification of the strains. You showed a very in the slide a very nice example where, depending on the protocol you choose, you can get a very different metabolic profile, so different result. So the protocol you decide to adopt is a critical step in your analysis. But I wonder which is the role of timing. So which is the bias you can have in the identification of a bacterial strain if you incubate the plate 24 hours, 28 hours, or even more? Do you see a significant effect, or it's stand more or less standard?
Yes. So in the manual of BIOLOG, the identification obtained, I don't know, in 24 hours is the best. Yeah? But I show on the presentation that I obtain two different results, and the results from 45 hours was okay. Yes. So sadly, we have to check for 48 hours and for 24 hours. So this is the cons of our tool. Yes.
Yeah.
But Okay, but we use two different. Okay, for identification, we use molecular biology and BIOLOG, and we compare these two data. So
I was curious because I never used BIOLOG for that. So yeah. Okay, thank you. I think we are almost to the end of this first day. So I thank again all the participants and the speakers. Today we will continue with the presentation on the filamentous fungi made by Flavia, and some insight on, let's say, linking the phenotype of bacterial strains with the genome, and how to link this information with the metabolic pathways and connect. I will show you some example with the co-inoculation of strains, and we will talk about data analysis and how to analyze and interpret data in a different way with some examples. So there will be more practical information and a lot of additional information for you. So thank you again. I don't see any final remark or question. So thank you so much. I see.
Thank you. Have a nice day. Goodbye.
You too. Thank you.
Thank you, everyone. Thank you, Stefan, for organizing this.
Thank you to you for joining us.
Well,
Again,
we'll
good
see.
contribution.
I'll see you tomorrow. I'm interested, of course, in your
Yeah.
presentation tomorrow.
Thanks.
Thank you.
Bye-bye.
Bye.